Hi, welcome to the pre-lab lecture for the Chlorinity experiment for Chem 100A. So in the last experiment, you measured the density and salinity of seawater, where salinity represents the concentration of all the ions in your seawater. But going forward in this class, we're going to start zooming in on other components, more specific components of seawater. So today we're doing chlorinity. And what is chlorinity? Definition. One common misconception with this term chlorinity is that it represents just chloride ions in solution, but in fact it actually represents all halide ions in the seawater that can be precipitated with silver nitrate. Sorry. So all halide ion concentration. Okay, halides, remember, are fluorine, chlorine, bromine, iodine, and now we're looking at ions. So it's fluoride, chloride, bromide, iodide. But I did mention that we're only looking at the halide ions that can be precipitated with silver nitrate, and fluoride does not precipitate out with silver nitrate. So we're just gonna ignore this guy. Okay, so chlorinity, we're looking at those concentrations of chloride, bromide, and iodide all combined. Now, one thing to note is that these occur in different concentrations in seawater. So as a proportion of the salinity, the total ion concentration in seawater, chloride ions make up 55.30% of that, whereas bromide, much smaller concentration, 0.19, and iodide, even smaller. And just for future reference, fluoride is also present in a very small concentration. Okay, so most of our chlorinity does in fact come from chloride, but just make sure you understand chlorinity actually represents all of the halide ions. Now, why might we care about this? Concentration of halides is important to be able to measure. If we just look at chloride itself, one important application in biology is osmosis. Concentrations of chloride is important for regulating cell size with osmosis. Things like if you took a saltwater fish and dropped it into fresh water, that equilibrium of the osmosis in their cells would be disturbed greatly, and all of the salt inside their cells would suddenly leave and their cells would shrink down and die. Or alternatively, take a freshwater fish and put it into seawater, and now all of a sudden that salty water is going into their cells and their cells explode. Okay, so osmosis is one important thing. Another application of chloride ion concentration being important is your lung cells transport chloride in a very particular way, and there's a condition called cystic fibrosis where if your lung cells are not transporting that chloride properly, that means you're not making mucus properly, and you have a tough time. Okay, so that's some applications of where chloride ion concentration is important and why it might be important for us to be able to measure it. Okay, so the method we're going to be using in this experiment is a titration. And so let's just think about titrations from the perspective that we all understand it, from a Gen Chem perspective. You may have be familiar with doing a titration with a burette, where you fill your burette with your titrant and slowly add it into your analyte solution until you have your moles of your titrant are equal to the moles of the analyte. Okay, so moles of titrant equals to moles of analyte is called the equivalence point. In practice, however, you typically add an indicator and you stop your titration once you hit the end point, which is when your indicator changes color. Okay, so that's one big takeaway from the concept of titrations, is that the endpoint is not equal to the equivalence point. The endpoint just represents when the indicator changes color, and that typically happens after the equivalence point. Okay, so we're going to talk about a bit later how that's going to cause a systematic error, where you're always going to stop your titration at the endpoint, but that's consistently giving you a result that's larger than where the equivalence point actually is. Okay, now some things I mentioned, this is kind of the general chemistry perspective on how to do a titration. But now we're in Chem 100A, we are analytical chemists now, we can do better. So what are some changes that we need to make? One thing that we do is, instead of using a burette, we're going to use a modified pipette. Okay, in titration where you use a burette, the drop size is very big, and by using a modified plastic pipette, you can get your drop sizes much smaller. And maybe you remember from doing titrations when you would add one drop out of your burette and all of a sudden your color would change drastically. But the smaller your drop size is, the closer you can get to an accurate determination of your endpoint. Okay, so using a modified pipette allows us to make our titration a little bit more accurate. Also, in a Gen Chem titration experiment, you probably used that burette and just recorded those volumes as you went. But in analytical chemistry, we want to use a technique that has more significant digits attached. And a burette usually can read to 2 decimal places in milliliters. But if we use an analytical balance, we can get 4 decimal places measuring in grams. So that's why in this experiment, instead of measuring volume, you're going to measure the mass of the titrant that you've added. Oops. Oh, sorry. Okay. Now, specifically for this experiment, what is the titration that we're doing? It's called the Fagens titration. So the reaction we're doing, we're taking all of the halide ions in solution in that seawater. I'll just call that X minus, and we're going to add silver nitrate. And that's going to cause a precipitate to form. Okay, so note that this is our analyte and this is our titrant. We're also adding an indicator called dichlorofluorocene. Okay, this indicator has some negatively charged functional group, so it's polar. So I'm going to call it IN minus. Okay, so just keep in mind that a negatively charged functional group will be attracted to positively charged ions. And that's going to be important for just understanding how this titration is happening. So let's just think at the very beginning, you have a solution of seawater. There's a bunch of chloride in there. And you add your first drop of titrant. You add your first drop of AgNO3, and it's immediately going to react with any halide ions in there and form a precipitate of AgX. So let's say, for example, there's a chloride and it reacts with that titrant and we form a silver chloride, AgCl. Now, at the beginning of this titration, there is a lot of halide ions, just excess. They haven't been reacted with yet. So we're just going to have a couple of those floating around. Okay, so this is what it looks like at the beginning. Now, as you keep adding more and more titrant, you're using up all of the analyte in solution. So you start having less and less chloride ions surrounding your precipitate, and instead, once you reach your equivalence point, all X minus used up, then you have you'll have excess silver. Okay, once you get past your equivalence point, you're going to start adding more titrant, and it has nothing to react with. So eventually, you'll have a system where you'll have your silver chloride, but now it's just getting surrounded by more silver ions that you add that have nowhere to go. Okay. Now, also, at the beginning here, your indicator is yellow. The indicator has nothing to attach to. You know, it's negatively charged. All those chlorides are also negatively charged. There's nothing really going on there, and it looks yellow. But now, at this final state, we have extra positively charged silvers, and those are going to complex with the indicator. Okay, complex. And this new complex is going to look pink. Okay, so when you do this titration, you're going from yellow to pink. You may remember in, say, a titration where you use a phenolphthalein indicator and you're going from clear to pink, you are aiming to get the very, very light, faint pink color of your indicator. You're looking for that in between of the two colors. So here's the same thing. You're trying to get just the faintest pink color in your solution. Some other things to think about with this procedure, you also add dextrin. Why do you add dextrin? All this coagulation is happening in the solution. You're forming a precipitate, and you want to make sure that your titrant can access all, everything in the solution. So we want to avoid any coagulation that causes any of our halides to get trapped. And dextrin is an anticoagulant, so that's going to help keep all of the ions floating around and free. Another thing is you add DI water to your analyte. And the whole reason you add the DI water is just so that your solution has a large enough volume for you to work with. And it doesn't really matter how much you add, it's not going to affect your concentration of chloride. Okay, now there is that one problem that I mentioned. Oops. Okay, our problem is that the endpoint and the equivalence point are not equal to each other. Okay, so this is going to cause some systematic error, some bias. But what we can do, it's going to be called a single point standardization. What we'll do is we're going to do our titration on our sample seawater, but we will also do it on a reference solution, a standard seawater solution. And this reference solution is going to have known salinity. Okay, this reference solution, so Scripps, they develop this very special reference solution that's used across the world for people analyzing anything related to the ocean. And it's a solution that has the same matrix as seawater. It's considered the standard of seawaters, and people have done all of these experiments in the past to figure out all the different constituents of seawater, and they make this reference stuff. They sell it in little bottles for a lot of money. So again, bottled water industry making bank. So standard SIO seawater. Okay, but note, we know the salinity of this solution, so now we need to make some sort of relationship between salinity and chlorinity, right? So map. Based on the percentages that I showed above, I'm going to scroll back. Okay, based on this idea that chloride, bromide, and iodide represent a certain percentage of the total salinity, we can create a ratio. Okay, so if we make a ratio of salinity versus chlorinity, it's going to give us the value of 1.80655. Okay, so we did our titration with a reference solution, so we can write that equation using those reference values. We also did the titration using our unknown, and that ratio will be exactly the same. That's just a constant. So unknown, unknown. Okay, so we can set those equal to each other, and now that's how we use our reference solution to connect to our unknown, and also using that known salinity of the reference, we can connect back to chlorinity. Another thing to note is chlorinity it, like we defined earlier, it's just the concentration of those halide ions, and concentration can be written in grams per kilogram. So this is grams of halide ions per kilogram of the entire solution. Okay, so you could calculate that directly for each titration, essentially, where the only thing you know from your titration is your grams of titrant, the grams of sodium of silver nitrate, and then you use the molar mass of silver nitrate to get moles of silver nitrate, and then you use the stoichiometric ratio in this titration reaction in order to find the moles of halide ions, and then you use the molar ratio of halide ions to get grams. Okay, so keeping that in mind, we can combine this first equation that I gave and create this new formula that is essentially all you need to use in this experiment. So the chlorinity of our unknown will be equal to the salinity of the reference divided by 1.80655 multiplied by some ratios here. We're going to take mass of the titrant for the unknown titration and divide that by the mass of the unknown. Okay, and same goes for the reference solution. Mass of titrant for the reference titration divided by the mass of the reference solution. Okay, so that's the formula you'll want to be using in order to calculate chlorinity of all of your samples. Now remember air propagation too. Oops. This is all multiplication and division, so you'll want to use that multiplication division version of the air propagation formula. Again, you just want to look at all of the terms in your formula, all of these variables. Notice, do they have an error associated with them? Okay, so things like the mass of the titrant for your unknown titrations. You're going to do 3 trials, so you should have an average mass, and there will be a standard deviation associated with those. Same with your mass of your unknown. And don't forget to keep in mind the tolerance of your instruments. Always take the larger number, the larger error. Okay, last thing to think about, just a few extra notes. First off is drop size. I already talked about how we're making a huge improvement by using a modified pipette instead of a burette, but still, how does that drop size affect your precision on your answer? Okay, so you should all think about that drop size. You will need to calibrate. Okay, when you are done with your titration, you need to figure out what the mass of one drop is and see how that mass is compared to the total mass it took for you to reach your endpoint and how great of an effect it might have on your error. As per usual, in your lab report, you're going to want to talk about error. Always break it down into systematic versus random. And remember, when you talk about systematic error or how accurate your experiment was, you want to look at percent error. And for random error, that's precision. You want to use percent relative standard deviation to make any arguments about how precise your experiment was. Okay, so those are the main things to think about with this chlorinity experiment. And now after this, you'll know the total concentration of halide ions in seawater. Thank you. Have a good